Welcome to the second part of the lecture 9 on the translocation of the phloem. This lecture is part of the plant physiology subject, which is offered in 2 degrees, the agricultural and the viticultural degrees offered at the institution of NMIT. Please refer to the recommended reading from Tays and Zeiger chapter 10 for complementary information, and ensure that you have watched both topic 5 on plant solutes and part 1 of this lecture before continuing. My name is Dr. Nikki Cooley. In this part of the lecture we are going to concentrate on loading and unloading of the phloem. We are then going to complete this topic by talking about allocation and partitioning. The image on your screen shows a leaf and the direction of translocation or movement of substances in the phloem. Sections of this leaf, as shown in figure A, were taken and examined under a technique called confocal microscope objectives. This allows a more in-detailed view of the phloem. Phloem loading is the process where photosynthates move from the chloroplast into the phloem. In this process there are several steps. The first step is triose phosphates. These are transported from the chloroplast to the cytosol and converted to sucrose. Other transport sugars are later synthesized from sucrose. The second stage is sucrose moves from the producing mesophyll cells to the cells near the sieve elements in the smallest leaf veins. This is short distance transport. And the third step, the phloem loading, where sugars are transported into the sieve elements and companion cells. Sucrose is then translocated away from the source, export, to the sink, long distance transport. Please familiarize yourself with this process. In the first lecture on solutes, we learnt about the different processes of aplastic and symplastic transport. Here the same philosophies can be applied. Phloem loading can be either aplastic or it can take a symplastic route. The initial short distance pathway of solutes is probably symplastic. However, sugars might move entirely through the symplast, or they may enter the aplast prior to phloem loading. Several mechanisms are now recognized, with aplastic pathway being the most common in this mechanism. The image on the slide gives you a visual representation of symplastic loading. If you start with CO2 and see how it enters the mesophyll cells. Sugar is then moved from cell to cell, as you can see when you follow the arrows in the diagram. The sugar moves from the mesophyll cell across the membrane into the plasmodesma, across another plasma membrane, where it eventually reaches the bundle sheath cells, then into the companion cell, and then into the sieve element cell. The image on your slide shows aplastic loading. You will note that the first movement of sugar from the mesophyll cell through the plasmodesmata, through the plasma membrane and into the bundle sheath cell is exactly the same route as in the symplastic loading. It is post the bundle sheath cell where this route changes. The compounds enter the parenchymal cells and the original ordinary companion cells, and then into the sieve cells, some through membranes and some over membranes, or in between the membranes. An important mediator of sucrose loading is a molecule called the sucrose H+ importer. It is thought to transport or mediate the transport of sucrose from the apoplast into the sieve elements. You will see the figure on the slide shows this transport. This mechanism uses energy generated by the proton pump. Energy dissipated by protons moving back into the cells as coupled to the uptake of sucrose. You will note the role of ATP and conversion of ADP in this proton pump. Now let us look at phloem unloading. This is the process where assimilates leaves the phloem. It is important into sinks such as developing roots, tubers and reproductive structures. Again, there are 3 steps. The first step is phloem unloading, the process by which imported sugars leave the sieve elements of sink tissues. This is then followed by short distance transport. After unloading, sugars are transported to the sink cells by means of a short distance pathway. The final stage is storage or metabolism of the sugars, depending on their function at this point. Allocation and partitioning are the most important functions of the phloem. Regulation of the distribution of fixed carbon into various metabolic pathways is termed allocation, while partitioning is the differential distribution of assimilates within the plants. Allocation includes storage, utilization, and transport of assimilates. Carbon fixed in a source cell can be used for either synthesis of storage compounds, starch is synthesized and stored within chloroplasts, mobilized for transport during the night, or metabolic utilization. This used within various cell compartments of the cell to meet energy needs or to provide carbon skeletons for the synthesis of other compounds. And thirdly, the synthesis of transport compounds. This is incorporated into transport sugars for export to various sink tissues. Can also be stored temporarily in the vacuole. Let us now look at partitioning in more detail. Sinks compete for exported photosynthates. This determines the distribution of transport sugars among the various sink tissues of the plant. Translocation of the sink tissues depends on the position of the sink in relation to the source. For example, young leaves might compete with leaves for photosynthates. Competition has been shown by numerous experiments in which removal of a sink tissue results in increased translocation to alternative, hence competing sinks. Conversely, increased sink size, for example increased fruit load, decreases translocation to other sinks, especially the roots. I have included the following URL link, which is a YouTube video on plant nutrition and transport. Please ensure that you watch this at the end of this lecture. This brings together some of the concepts we have been talking about in this lecture. In summary, after watching parts 1 and 2 of the translocation of the phloem, I hope that you have some understanding of the translocational pathways, the phloem function, the pressure flow model and how it explains movement in the phloem, solutes in the phloem, the structure of sieve elements, companion cells, sieve plate pores, the processes of both phloem loading and phloem unloading and the 3 associated pathways for each, and that you have discussed and understood allocation and partitioning. Please ensure that you read the chapter in the Tays and Zeiger to familiarize yourself with some additional detail for these sections.